Welcome to our latest installment of the GI Community Chat, and thank you very much to both of our guests for joining me today. Joining me are CDI's partners from the Functional Gut Clinic is Dr. Anthony Hobson, as well as from Owlstone Medical, their CEO, Billy Boyle. Welcome, Anthony and Billy. Thank you very much for being here.
Nice to be here.
Today's topic of conversation is around trace gases and breath testing for gastrointestinal disorders, its current state of play, its future outlook, its clinical interpretations. This is going to be a really meaningful topic of conversation. It's one that I personally have been looking forward to having for quite some time. It's also an important one as diagnosis, treatment, and medical care in general becomes more and more personalized and precision-based by the day. Disorders of the gut-brain interaction or functional GI disorders, as they were formerly called, are at the top of that list. And many of these patients are motivated to find a care plan that works for them. And with the growing number of diagnostic and treatment options that can be individualized to address their own unique pathophysiology, I think this future looks promising with regard to being able to both qualitatively and quantitatively find some answers to some of their digestive distress, which brings us to the topic of discussion today and the truly amazing work that's being done by our partners in the UK at Owlstone Medical and at the Functional Gut Clinic. Hydrogen and methane breath testing to aid in the diagnosis of some of the common sources of digestive distress like small intestinal bacterial overgrowth or SIBO, intestinal methanogen overgrowth or IMO, carbohydrate malabsorption is well-established means of clinical use. However, it's by no means a perfect one. The need is clear and apparent to continue kind of expanding upon those established markers in breath, i.e. methane and hydrogen, and pinpoint the diagnostic strategy even closer, make it a little bit more precision-based. This is where the fascinating new frontier of volatile organic compounds and short-chain fatty acids can really make a distinction with the difference and the opportunity to specify the true etiology and pathophysiology of disease in these functional GI cases. Already that data speaks pretty significantly. There's meaningful implications of being able to quantitatively analyze the levels of hydrogen sulfide in breath, for instance, and this is just the beginning, but there's also some serious considerations to account for topics like methodology of analysis, i.e. whether it's using gas chromatography or select ion flow mass spec, limits of detection, whether you're getting down to parts per million or parts per billion, sensitivity and specificity of the test itself, study design, sample collection and transport method, and so much more all play a major role in ensuring the clinical integrity and validity of the test results. So these are all areas that we have been all working together to get right. And so without any further ado, I think I'd like to turn it over to our panelists and to CDI's partners. And I know they will have a lot more to say on all of these topics, but I figured I'd start with you, Dr. Hobson, to perhaps give us a little bit of that state of play and general overview of the current outlook on hydrogen and methane breath testing and functional GI disorders, as well as how you see the role of hydrogen sulfide specifically, but then more generally these short-chain fatty acids and volatile organic compounds playing into the overall care cycle for a functional GI case. And I know you're on the front lines seeing these patients on a daily basis. So both from an evidence-based perspective and anecdotally, I'm sure you have a lot to share there. And from that point, I think we can turn it over to Billy to maybe give a little bit of an overview on the new applications for these compounds and all the fascinating work that's being done at his lab in Owlstone in the UK to substantiate these new biomarkers. So with that, I will stop talking and turn it over to you, Dr. Hobson, to please share some of your thoughts on any or all of the above.
Thanks, Craig. Great to be here. Great to be having this debate as well. As you know, we've been using hydrogen and methane breath testing in our clinic for about 10 years, and for the past 6 years in partnership with CDI, as we felt that the CDI methodology was the most sensitive and robust and gave people the flexibility of doing testing at home, for example, or in the clinic. And what we've really been focusing on during that period is to try and make the current established methodology better. And what I mean by that is by using the data that we collect to inform us about how important that data is. And that's in collaboration with the people who refer to us, but also there's a whole community out there that are also doing really valuable work. And I think it's quite rare for a test to have such specificity in terms of a biomarker of microbiome function as a hydrogen methane breath test. So, these gases are only produced by the gut bacteria. So, by definition, if you add a carbohydrate probe and you begin to pick these gases up, then it's something to do with the functionality of the microbiome. And that's exciting in itself. It's a good starting point. But then when you look at as well that biomarkers of hydrogen and methane are also specific to response to treatment, then it becomes even more important. So we know that if you have a positive hydrogen test for SIBO, you're much more likely to respond to a treatment like rifaximin. So you get a 60 to 70% response to rifaximin, whereas if you have a negative breath test, you maybe only get a 20%. And when you consider the cost of treatment and the implications of treatment, that's a massive healthcare benefit for both patients and also a cost saving. And it's the same with EMA, with methane. We know that if you improve the methane profile, you improve constipation and you improve the clinical picture. So we already have a really great test that's robust, that's repeatable, and predicts response to treatment. But there's still some gaps in there. So as well as making sure that what we're doing is as robust as possible, what we also want to do is try and learn how we can make it better, but then add things into the equation that will give us even more sensitivity in the future. And I think, there's also been this debate around small bowel and colon. Now, to me, it's always been a moot point because we are just as interested in the last part of the breath test as we are with the beginning of the breath test. So what I mean by that is what's happening in the small bowel and what's happening in the colon. And there's data going back to the 80s that from John Hunter at Addenbrooke's, who coined the phrase colonic malfermentation and showed that if you get malfermentation and dysbiosis in the colon, then you'll get an increased production of gas, and this responds to treatment, and that treatment also then gives people symptomatic benefit. So what we want to try and do with the future now for breath testing is kind of elongate the offering. So we have a small bowel profile, we have a colonic profile, and then we can start to add on other biomarkers that we think are important. And hydrogen sulfide's had quite a lot of press recently, and that certainly is an important molecule or gas. It's associated with pro-inflammatory states. It's associated with diarrhea, inflammatory bowel disease. So I think that's an important tool that we've worked hard on to try and measure that. And I'll let Billy talk about some of that work shortly. But there's thousands of other compounds that are being produced by the gut microbiome. And at the moment, we have to rely on stool samples to see what the microbiome is doing, which is messy, and it's the kind of the stool is coming out of the end pipes, it's the waste product. We're actually, what we can do with breath testing now by looking at the later time period is to look at these thousands of compounds that are being exhaled on breath that will give us a fingerprint of microbiome functionality in a broad spectrum of patients. So I think that's where the future lies for us. And, we can talk about some of the work that we've done to try and validate that later.
Yeah, I think you bring up a multitude of really important and fascinating points. The one key takeaway I have from that, Anthony, is the idea of this being a non-invasive means of diagnosis as well. So, the gold standard being a jejunal aspirate for small intestinal bacterial overgrowth, as we all know, is just not a practical means of diagnosing a functional GI patient, and to sedate a functional GI patient who is absent alarm signs and put them through an invasive procedure such as an aspirate to diagnose SIBO is not something that's being deployed clinically very often. The breath test is a very useful, substantiated, and validated means of being able to diagnose that same disease. But to your point, the two trace gases that we have today and the third in hydrogen sulfide is really just the tip of the iceberg. And I think as we dive deeper, it's important to recognize that not only will we be able to clinically validate these biomarkers further, but they'll actually be readily accessible and able to meaningfully diagnose patients clinically, as opposed to just having potentially a procedure-based pathway where you're not going to be able to get a meaningful diagnosis on as many patients who have been suffering from this disease. On average, an IBS patient, which, there's data out there that shows up to 80% of IBS patients have SIBO, for instance, suffers 6-7 years before they get a meaningful diagnosis of irritable bowel syndrome. Many times it's empirical because the provider hasn't done a procedure or a diagnostic test. So I think as you continue to add biomarkers and validate them to a test menu that can be done on the same test, in addition to a hydrogen, to a methane, to a hydrogen sulfide, you're only going to be able to make that care conundrum that much more precision-based, that much more individualized, which is ultimately the name of the game here. And I think that is a really good opportunity to segue, Billy, maybe into some of the work that you're doing at Owlstone on hydrogen sulfide. Truly fascinating, truly important. And I think we're getting it done, and we're getting it done right, which is exciting for us to watch in the background.
Yeah, thanks for having me. Really excited to be working in our collaboration and partnership. And I think, as you said, there's really important problems to solve here, both from a patient perspective, but then fundamentally making sure the technologies work effectively in terms of sample collection, analytically as well. So I thought I might just give you a 30-second background to the history of Owlstone as well, just for your listeners. So we're originally a Cambridge spin out. So we spun out of Cambridge back in 2004 and developed miniaturized spectrometers. So mobility spectrometers on a chip. And you can functionalize it to look for a range of different gases. So we were kind of thinking about how can you apply this technology. Interestingly, hydrogen sulfide popped up quite a lot in non-medical applications to start with. So things like industrial health and safety, toxic industrial chemical monitoring, those types of things as well. So we had a lot of experience there in this adjacent field. And as we grew in Owlstone and matured these technologies, not just for analysis, but also sample collection, we started to get much more interested and serious about breath gas analysis. I think, as both of you touched on, we know about hydrogen and methane today, hydrogen sulfide, but we know as well that there are thousands of volatile organic compounds on breath. So what we did is spin out a separate business, Owlstone Medical, that's really trying to do two things. The first is develop valuable breath-based tests that address real clinical problems and do that kind of directly, and then also trying to work with academic, clinical, and pharma researchers to try and understand the full breadth of the molecules that are on the breath and understand what they relate to. What's the underpinning biology for these compounds. So we've developed an approach that allows you, I'll hold it up to the camera, hard to see through the camera, but we kind of divide the world into the sample collection and then the analysis. And it's crucial, you need to get both right. So we know in hydrogen methane testing, there's external tubes, bag-based sample collection. When you go slightly broader than that, and think about VOCs, volatile short-chain fatty acids, and it becomes much more important and difficult to get a good sample. And that's what we're able to do with this device here, this receiver device. So a subject can breathe into the device. We look for different fractions of breath, and you can select specific fractions where we then activate pumps to draw VOCs down into sorbent tubes. And the sorbent tubes strongly bind the volatile, so it stabilizes it, which means you can then analyze later by heating the sample up and releasing the volatiles. So I think when you look at this, untapped and very diverse range of molecules, that type of approach makes a lot of sense. We've obviously been working quite closely together in some studies looking at these short-chain fatty acids and other microbiome-related compounds as well. And there's some exciting posters and additional papers to come from that work as well. We've also been looking together, as you know, on the hydrogen sulfide side of things. And again, I think you need to get the fundamentals right. How do you ensure you get a good sample? How do you ensure you can analyze correctly? So a lot of the work that we've been doing is focused on using things like PVDF bags, ensuring that you have stability over long periods of time when you're transporting samples around. And then also looking analytically, what are the right types of analytical approaches there to measure gases such as hydrogen sulfide. So there we use selected flow mass spectrometry, which is basically different flavor mass spectrometry. And then when we look at the broader range of VOCs, we use high resolution accurate mass, so GC Orbitrap mass spectrometers from Thermo. So I think fundamentally we believe that you need to get the core platform right, really pay attention to those dual challenges of how do you get a good sample, how do you have robust analysis. And that then gives you a valuable tool to go out and run clinical studies, find what normal ranges look like in human breath, and then start to apply that within different disease populations as well. So really building it up brick by brick of what is the potential of breath above and beyond what is known today with hydrogen and methane.
Yeah, and Billy, you touched on a few really key points there. I'd love to talk a little bit about the volatility of hydrogen sulfide, and you mentioned the word fundamentals. I think that is so important. Just even getting down to the bare bones, the true basics of this, whether or not you're analyzing an otolaryngal sample, a true alveolar breath sample, or nose exhaled air, can make all the difference in the world on the outcome of that test result. How that sample was collected, what the means of transport was, temperature control, the methodology that you're using. If you wouldn't mind maybe just touching a little bit on just the volatility of hydrogen sulfide in general. I mean, there are technologies in the market today that I think are marketing to quantify hydrogen sulfide. I'm not sure what methodology those tests are being analyzed on. I believe the limit of detection is in the parts per million range. But if you wouldn't mind just kind of drawing the distinction between the two, why it matters, why collecting mouth over nose air matters, why making sure the sample collection device is truly capturing the volatile organic compounds and maintaining their stability and integrity during transport, so you're actually testing a true human breath sample at the time of detection, as opposed to any atmospheric dilution that may have crept its way into the sample during transport and the like. Just some of those quality control mechanisms and assurances, I think, are really, really important in this regard, because once you get out to market and start offering a test commercially, you want to make sure that it is as clinically valid as you possibly can. I mean, obvious statement there, but not necessarily always practiced.
Yeah, and I think this is where a lot of our previous heritage in VOC and gas detection uh, is really advantageous. So, back when we were looking at hydrogen sulfide within the context of industrial health and safety, so exposure of the gas to humans, we've developed a lot of test capability that allows you to do things like generate, traceable, gas concentrations traceable back to primary standards. So that allows you to start to do things like, test different sample collection approaches, ensure you have stability, start to increase complexity by blending it in with other gases that you find in human breath, other matrix compounds, blend in humidity and the like as well. So I think it's really, again, taking that step by step approach and evaluating the data at each stage there. And then, once that is established, then you're looking more at the true value of what is coming out of the human subject that does have obviously some of these background VOCs plus humidity as well. And I think the same holds true on the analytical side. So I think Anthony might touch on some of the observations that have been made in terms of the concentrations that you're saying in normal healthy subjects. So I think what we've focused on with the SIFT-MS approach is ensuring you have limits of detection down at that low ppb, tens of ppb level. I think that's where you need to be operating analytically. So again, I think it's just been systematic, looking at where sources of pre-analytical and analytical error can creep in, and making sure you do enough testing so you have confidence in that end-to-end workflow from sample collect to analysis.
So important. Yeah, great points. I appreciate that. And Anthony, going back to this idea of data and the interpretation in and of itself, I'd love to get your thoughts, just clinically speaking. We know hydrogen is produced by microbes in the gut. We know that methane is produced by archaea in the gut. We know that hydrogen sulfide or sulfate reducing bacteria can gobble up both. And so, patient presents with a flatline breath test, but is symptomatic and displaying signs of SIBO, is absent alarm signs, or displaying signs of functional GI disturbance, I guess I should say, IBS, whatever the case may be, absent alarm signs. In a case like that, if you were to order a hydrogen sulfide test, what are the characteristics of that test that you're looking for? What would make you confident as a provider to know that the result of that test is one that is clinically meaningful to you, or are you empirically just moving in the direction of treating that patient as SIBO and assuming that it might be, sulfate reducing bacteria or hydrogen sulfide that's gobbling up that hydrogen and methane?
So when we get a flat line, it can be caused by two things. We know that people with SIBO have impaired transit quite often and motility. So because we're only looking at about two and a bit hour period, then it could be just that the substrate's not reached where it was heading. Or it could be that there is hydrogen sulfide present. So we may ask the patient, do you have, it's a great term, malodorous flatulence, this kind of eggy flatulence. Are you having diarrhea? Did they fit more of a hydrogen sulfide type profile? And then we could have a discussion with the clinician at the multidisciplinary team and say, well, given their symptoms, their profile, the flat line, they may be likely to have it. So you may want to go down the antibiotic route or some of the treatments that they might want to go with a sulfur, low sulfur diet, etc. Or you might want to do a transit study. So we quite often do a smart pill study on these patients, which again, is gives us a whole gut motility profile to go with that. But if we did or were able to have hydrogen sulfide on top in these patients, either as an adjunct afterwards or as part of a combined test, then even though it's not a huge population, maybe 10% of all the people that we do, it certainly would save time and effort and give us some solid decision making. And we have started now. We've profiled this in about 40 healthy subjects, and we've got a really good idea now of what the normal ranges are. So we're just starting to roll this out in a few select patients, and we're getting some really interesting data. I think it's, as I say, hydrogen and methane will be the mainstay of what we do, and whether we combine it or do it as an adjunct, we'll have to decide in the future. But it's all about, as you say, Craig, precision medicine, making sure people don't get unnecessary tests, making sure they don't get treatments that might actually make them worse and disrupt their microbiome.
Yeah, I think that's so important. It's been empirically diagnosed for far too long, at least in my opinion, especially when there are tests out there to be able to diagnose with some level of finality. And Anthony, you touched on another really interesting point of just being able to kind of categorize or better index these groups of patients or cases. And I wanted to talk a little bit about just kind of comorbidity here. There's a few studies over the last few years that have substantiated H2S producing cases as being more diarrhea predominant, IBS, if you will. Obviously, methane has been most closely correlated to constipation, not in all cases, but in many of them. But now there's a lot of overlap. There was a study, the name and author is escaping me, but we'll put it in the notes here. I think it was last year in the Red Journal that was showing a close correlation between hydrogen sulfide producing patients and inflammatory bowel disease. So, kind of crossing the chasm from functional GI and moving over into potentially anatomic disturbance. So if you wouldn't mind maybe just touching on one or either of those areas, within the realm of functional GI, how you see this helping to better index and categorize these patients to be able to expedite a treatment pathway that is precision-based and hopefully will solve their problem that much faster, but then also potentially using hydrogen sulfide or other trace gases in breath as a biomarker outside of just the core competencies of functional GI. There's a lot of direct correlation between just small intestinal bacterial overgrowth and other autoimmune disturbances. There's a lot of data in rosacea, scleroderma, the like. So maybe just generally or specifically how using biomarkers in breath can be used to better characterize these patients in functional GI and outside of it.
So I think the most important thing in science is to have a good model. And I think Billy has talked about getting the technology right and making sure that you've excluded as many variables as possible. And then what you're left with is a robust model that you can test by experiment. And this is exactly where we are now. We're at this exciting phase of look at new therapies. So if we can put to bed the argument about whether it's small bowel or whether it's proximal colon, because the two things are linked inextricably, and they're probably both going to be affected by most of the treatments that we give, be that antibiotics or diet or intestinal absorbents or whatever. So if we put that to say, right, we now have a model of microbial fermentation in the gut. Okay, so the model is robust. We know what it does. We know it responds to treatment. If you treat people, they get better. Then you can start to just gently manipulate different elements and different treatments and try and test that and see what works in different patients. Before then, we didn't really have a robust model for it, but now we have got this. And I think one of the problems for drug development in functional GI disorders has been that most of the endpoints have been based on symptoms. And you've got a high response, so you may have to get above 40% placebo response just to see efficacy. But if we can go to a biomarker-driven model of functional GI disorders, then we can start to see which treatments, we can compare treatments. There's no shortage of patients out there. So I think the next generation for me for developing things in functional GI is to start to collect the data in the broader patient population. And one of the things that we're doing sort of later this year is to work with different companies who've got products that might not be pharmaceutical products. They may be probiotics, they may be prebiotics, they may be intestinal adsorbents, things that are available to functional GI patients to purchase. And you know how big the market is, especially in the US with supplements and things, but it's a market that isn't very well regulated. But we can use this robust model to feed in different treatments and get objective evidence of what actually works, how it works, and what's the best benefit for these patients. So that to me is like one of the most exciting parts of this research collaboration. And the second you touched on with IBD. So IBD, quite rightly, the focus is on controlling disease. But the drugs are pretty good for that now. So when these patients are in remission, what are they left with? Well, they're either completely happy or they have a functional GI overlay. And the functional GI overlay is the thing that reduces a patient's quality of life significantly. So they get fear of eating, they feel miserable, they stay off work. And yet, because they're in remission, that kind of aspect of their condition is not really looked at. And I think this is really important that we, work with other collaborators now to look at this kind of microbial profile in IBD patients in remission and see which treatments will be effective for them as well. I know there's been a big call in the IBD world in the UK to look at some of the core elements of that. And one of the big elements is abdominal pain in patients with remission. So what causes it? Is it dysbiosis? Is it gas production? Is it dysmotility? So I think on the functional GI disorders, we've got a model we can test by experiment. In IBD, we need more of a fact-finding mission to look at the panel of biomarkers in an IBD patient, see if it's different, and then see if we can have an equally robust model that we can test with different treatments in those patients.
Yeah, I love this idea of a true microbial profile and using a biomarker-based approach to diagnosing and treating, starting with these functional GI patients. But completely to your point, Anthony, many times an IBD patient is also a functional GI patient, but that secondary set of afflictions may well be discarded altogether because they're on drugs for IBD or whatever the case may be, and they feel like or have been told they are in remission, but symptoms prevail by virtue of something that is pathophysiologic in nature and not necessarily anatomic. But using these biomarkers to get there, I think, provides a clear path. So, Billy, maybe kind of taking us to the home stretch here. Some of the work that you are continuing to do in this field at Owlstone, your team in the lab and these biomarkers, anything on the horizon with hydrogen sulfide, any new data, prospects for commercial launch with functional gut clinic on the hydrogen sulfide side, and then any potential new VOC work or short chain fatty acid work that you guys have in the pipeline that we should know about?
Yeah, I think we're getting very close in the hydrogen sulfide side of things. And again, I think for all of us, it's making sure the data is there to support it. So I would say watch this space in the very near future. And I think you're building on the point that Anthony made as well about the broader range of potential biomarkers. So some of the work that we've been doing at Owlstone is building a VOC atlas So again, trying to characterize everything that's present on the breath and origin and normal ranges, etc. And I think when you look at the thousands of volatiles on the breath, and you ask the question, where do they come from? So some are produced locally within the airways, which makes sense. But we know already with hydrogen methane, gut to blood, out onto the breath is a route. And actually, when we've looked at all the molecules that we've characterized are in the VOC atlas, you get the respiratory origin ones, quite a lot which are related to the liver. So where you have exogenous substrates coming in, which are being metabolized in the liver, and you see patients who have liver disease that impacts how they metabolize some of those exogenous substrates. But most of them, actually, we're saying are in one way or another related to the microbiome, so gut origin. So I think what's exciting is, and I think we'll be able to put it in the show notes as well. So we've already been able to characterize that you can detect a lot of these very interesting compounds, such as short chain fatty acids, where studies have been done looking at how they change through things like iron supplementation. We've done studies with the Mayo Clinic looking at exhaustive exercise and how that changes levels of acetic acid on breath. So I think because that measurement tool now exists, I think that unlocks a lot of really quite interesting new science and studies. And as Anthony said, there's a lot of potential interventions that can be tested. And I think, again, breath offers a really unique set of advantages. We've done a lot of VOC work looking at headspace of fecal samples, urine samples, etc. But as Anthony said, you get a fecal sample, once in some period of time, and that limits what and when you can interrogate from a VOC perspective. But with breath and the sample collection here, you can collect sample anytime, any place. So it allows you to get this very rich data set following the time course, where again, you can look at VOCs being produced by the microbiome or put in new novel substrates as well. So again, this idea of test and response to treatments, or putting in specific substrates that target specific components of the microbiome. I think all of it is now possible. So I think obviously with our collaboration, there's lots of fantastic new science and research that will come out of that. And I think importantly, from a patient perspective, it's going to help to identify what interventions work for them in an objective biomarker-driven approach.
I couldn't agree more. I'm excited about the collaboration. I think we all are like-minded in sharing the same goals here with patients in mind. And I'm looking forward to the continued work, and everybody stay tuned for updates and announcements regarding commercial launch of hydrogen sulfide, certainly in the UK, and more VOC work to be done in the future. So with that, I will say thank you very much to both Dr. Hobson and Billy for joining us here today on this GI community chat. And until next time, thank you all for joining us as well.